So I'll be doing this presentation from the perspective of genome integrity, because it's impossible to cover every aspect of nutritional genomics, and it's also an area where I've really focused most of my efforts in terms of research. So we'll be covering some of these topics here. What do we mean by genome health and disease risks? Some genotype-based diseases relating to DNA damage, and preventative effect of dietary bioactive compounds, and something about personalized nutrition in this area as examples. Okay? So let's start from DNA damage. So to begin with, the genome can be damaged in many different ways, such as DNA strand breaks, DNA adducts, small, bulky, and for each of these there is a particular DNA repair pathway to try and correct the insults, the genetic insults that have been accumulated or induced. There are at least 120 different genes. There's more than 120 in fact, that are involved in either sensing the damage or excising the damage, repairing it, patching it up, and so on. And I won't go into those details, but for example, in terms of oxidative stress, you're probably going to have some strand breaks, some base lesions such as oxoguanine, and the glycosylase in that case is OGG1 that recognizes the oxidized guanine and removes it. In the case of strand breaks, then there's a number of pathways, non-homologous enjoining and homologous recombination repair to correct these lesions. Now, why is DNA damage important? We know it's important because loss of genome integrity is central to many of the aging acceleration conditions due to genetic defects in important either the structure of the nucleus or in the repair process. And these are just some of the most striking examples, such as progeria, where there is a defect here in the lamin proteins. The lamin proteins are very important. They just lie below the membrane. They're very important to keep the nuclear membrane functioning well. When there's a laminopathy, when there's some defect here, you get a kind of a hernia in the nuclear membrane. And also important because there's an important interaction between telomeres and the and lamins, that's critical for telomere maintenance. In the case of ataxia telangiectasia, there's a problem here in the ATM gene, which is important in damaging and sensing DNA damage and then recruiting the required repair complex for it. In the case of the skeratosis congenita, there's a problem in telomere maintenance. There can be different kinds of lesions, such as defects in telomerase. It could be defects in the proteins, so-called Choterin proteins, which are needed for telomere function. And another example would be Werner's syndrome, where in this case there's defects in the helicases that are needed to work together with topoisomerases to recognize guanine quadruplexes that can emerge when DNA is in single-stranded form, and to resolve these, otherwise it will cause replication stress and generate other kinds of damages, including strand breaks. So for all of these reasons, we know that increasing DNA damage in various ways will accelerate aging. Now, DNA replication stress can be due to DNA damage and or nutrient deficiencies, which cause stalling of the DNA replication fork. And the bases, for example, that are needed, such as thymidine, which is generated from uracil, are really generated in a kind of just-in-time system to as DNA is being replicated. There's some very nice work by Patrick Stover showing this happening in the case of a synthesis of thymidine. But if there are any adducts here, that's going to cause stalling, it's going to cause some breakage occurring as a result, and this will, of course, create havoc in the ability to properly replicate the DNA. So nutrition plays an important role here in supplying cofactors for DNA synthesis. Now, there are many ways we can measure DNA damage in cells. This can be done either at the cytogenetic level, which is the classical way we used to measure DNA damage and still do. But this was the first way we really could measure DNA damage specifically and sensitively. Another marker is the micronucleus index. These were first recognized more than 120 years ago. This predates metaphase analysis. The cause of that wasn't known, but it was linked with anemia. And eventually it was found out that these micronuclei, or so-called Howell Jolly bodies, named after the people who discovered them, are induced by folate and B12 deficiency. But they can also be induced by chemical genotoxins and so on. Another way to look at micronuclei is to do that in lymphocytes using the cytokinesis block micronucleus assay, which I'll talk a lot about. It's a method I developed many years ago, and we use a lot in nutrition studies because it's very sensitive to nutritional deficiency. You can also measure DNA strand breaks using assays such as the COMET assay. One can also measure telomere length by PCR or other methods. You can also measure mitochondrial DNA deletions by PCR. Here's an intact mitochondrial DNA, and when there is damage, then you'll get major deletions, and these appear as bands beneath the larger band that you would typically see in intact DNA. And this kind of lesion you can see in tissues of older people, for example, in the muscle. If you look at the muscle of people older than 65, most of it will be the deletion form rather than the complete form. You can also look at base lesions such as atoxoguanine, as well as DNA methylation. So all of these are possible. They are well-validated methods, but some are better validated than others. Now, the earliest paper I could find of an association between the DNA damage and malnutrition is this paper, which I showed in the previous lecture on protein calorie malnutrition and chromosome aberrations. And what you can see here is well-nourished infants in black and the malnourished infants here in the white bars, and this really massive increase in chromosome damage, at least 5-fold higher. Okay? And these are levels of increase we would normally associate with let's say, what you would see with a significantly higher dose of radiation. So malnutrition causes as much DNA damage as ionizing radiation, for example. Okay, so the idea that malnutrition is not as harmful as radiation is a nonsense. It's clear that malnutrition causes serious levels of chromosome damage. Now, the other way measuring chromosome aberrations is quite complex. So the cytokinesis block assay was developed because this assay is done mainly in lymphocytes. Just from a drop of blood, you can do this measurement, but you have to stimulate the cells to divide and express the damage, like as you would need to do to look at metaphase chromosomes. And what happens here is the fragments of chromosomes can appear lag behind and form micronuclei, or if you have a dicentric chromosome, it will appear as a bridge, as you can see here. You can also have lagging whole chromosomes. I'll go in more detail. So the ratio, so you need to block the cells in the binucleate stage and then score these lesions visually, which is not difficult to do. Now, there is a protocol paper published in 2007 in Nature Protocols, which has all these details. You can go in more detail in this system. You can look at necrosis and apoptosis. And apart from the micronuclei and the bridges, you can also measure these nuclear buds, which are a mechanism indicative of gene amplification. This is a way that the nucleus eliminates excess DNA, okay, or unresolved DNA repair complexes. So that tells you there's genomic instability. And the fact these features, micronuclei, bridges, and buds, are essentially the image or the fingerprint of genomic instability, like you would see in cancer, for example. Then you can use molecular probes to see if there is you can even have a binucleated cell without a micronucleus, but you could have chromosome malsegregation, and you can measure that by molecular probes, for example, for chromosome 21, which is very relevant in terms of increasing the risk of Down syndrome, or you can tell whether a micronucleus has a whole chromosome or a fragment in it by a centromeric probe, okay, and so on. Now, dicentric chromosomes can occur due to misrepair of breaks. Okay, so you might have a double-stranded break in two chromosomes, and instead of restituting the fragment back to the original chromosome, you have a misrepair, so that the break end from one chromosome is joined to that of another, forming a dicentric and then a fragment. This can occur due to misrepair, in other words, because there is some defect in genes such as the ataxia-telangiectasia gene, the BRCA1 and BRCA2 genes, the breast cancer genes. This is why they are important and why they can increase the risk for damage, and RAD51. And you can observe this as a bridge in the binucleated cell, because the dicentric is pulled to opposite poles, because there's a centromere at either end, and the fragment appears as a micronucleus, and so on. And using these features, you can get some idea of the type of damage that is induced by the agent or the situation that you are investigating. Now, in fact, there are many types of chromosome aberrations, which I won't go into detail of this. There's not many people who are skilled anymore to be able to recognize the various ways of measuring chromosome damages, except for those who are involved in radiation biodosimetry. And that is the reason is that chromosome analysis, the dicentric chromosome, is one of the gold standard measures of measuring radiation exposure, as is, in fact, the micronucleus index as well in binucleated cells. And these are standards that are used in the International Atomic Energy Agency manual and WHO-endorsed manual for radiation biodosimetry. We know from a vast literature that these indices, chromosome aberrations and micronuclei, are sensitive to all of these exposures, from chemical genotoxins to social status, psychological stress, poor lifestyle, malnutrition, and radiation exposure. So when we use these biomarkers, we are in fact measuring the total, the exposome effect, not necessarily of just the agent or the condition that we are looking at. And this, while it is challenging to distinguish between what is the cause of the increased damage, it's also very valuable because it actually is giving you the full image of what is happening in the population or in the individual. Then it's a matter of teasing out what is the likely cause, what is the plausible cause. Okay, let's say in the absence of radiation exposure, obvious chemical exposure, what is the risk that you could look at? Could you look at the plasma, see if there's any nutritional deficiency that we know is associated with these biomarkers? Now, validation of biomarkers is very important. So now I'm going to focus on the micronucleus index and lymphocytes. So I'm going to focus a bit more so that we can go a bit deeper. And for every biomarker, apart from in the case of nutrition, you need to show an association with the nutrition, cross-sectionally, some deficiency or excess. The marker has to be sensitive to that. It's got to be sensitive to intervention. We need to have those studies to validate it further. Which nutrients can you intervene with that it responds to? And then we also need association studies with disease. But furthermore, we also need, ideally, prospective studies that show that the biomarker, when it is high, in this case of DNA damage, it actually is associated prospectively with increased risk of an illness. And I'm showing you here some 4 examples for the micronucleus index in lymphocytes. Here is a study we did in mothers at 18 weeks gestation who had previously had normal pregnancies. We tested the hypothesis that the micronucleus index at 18 weeks gestation might predict the risk for preeclampsia or intrauterine growth restriction of the fetus. And the study showed, it's a relatively small study, but it did show an association of increased risk for those with micronucleus index greater than, let's say, I think it was 32 per 1,000 cells. So that is one association. We know that this index has also been shown to be associated with infertility and risk of miscarriage as well. But this was in cross-sectional studies. Prospectively, we put together data from about almost 7,000 people collected from around the world, where the micronucleus index was measured, and it was possible to link with their cancer outcomes 10 to 15 years later. And what this study showed was that those with a medium or high level of micronuclei had about a 70% increased risk for cancer. Similar studies were done in relation to cardiovascular disease mortality, both in patients as well as those who had not developed cardiovascular disease at this stage. In relation to dementia and other neurological disease, we only have cross-sectional data so far, and that is data showing that the micronuclei are increased in Parkinson's and in Alzheimer's disease. And interestingly, in Parkinson's, the micronuclei increase is mainly due to the breaks in DNA, whilst in dementia, it's due to chromosome malsegregation or losses. We followed up some of these studies in dementia a bit further. In the study, we looked at the association of these biomarkers, micronuclei, bridges, and buds, with a cognitive test that is commonly used in mini-mental state examination. And what this study showed was there was a negative correlation of these markers in controls, mild cognitive impairment, and Alzheimer's, in that those individuals that were higher in these markers were more likely to have poorer scores on the mini-mental state examination. So there seems to be an association here, again, with DNA damage and cognitive ability. We also are now looking at some other markers which have not been investigated previously in nutrition that are of interest. And one of these, this is work done by our postdoc Maxime Francois, who joined us to start focusing on this question of G-quadruplex DNA. What he first did was to look in the same study where we were looking at mild cognitive impairment to see whether there was any effect of G-quadruplex. This is measured using a probe that identifies the G-quadruplex structure, and they also measured gamma H2AX, which is a marker of strand breaks in DNA. And essentially what he showed here is that G-quadruplex frequency was elevated in mild cognitive impairment. It's elevated both in terms of the frequency and the intensity of the signal, as well as gamma H2AX is increased also, and they are both correlated with each other. Furthermore, the G-quadruplex measure is correlated well with micronuclei, bridges, and buds. Furthermore, in vitro studies also showed that folate deficiency increases the frequency of G-quadruplex. And we know that folate deficiency increases micronuclei as well. So there seems to be a spectrum of damage that is associated with cognitive decline and with nutritional status. Now, I wasn't sure where I'm going to put this slide because I got to put it in 3 or 4 different places. But as a bit of a break, I'm now going to deal with the question of aging. So when I first joined our CSIRO, the nutrition division, I decided that what I should be doing is, having moved from a field of radiation biology and genetic toxicology and chemical toxicology, I was reasoning along the lines, well, if we are going to worry about chemicals and radiation, we should first understand baseline. What is the baseline frequency in the population who are not exposed to chemicals and genotoxins? And if there's any variability, is it related to nutrition? Right? So that's how it all started. And that started with a study on vegetarians and vegans, which I'll show, vegetarians and non-vegetarians, which I'll show a bit later. What was evident, now this is an accumulation of data from a number of studies, but first of all, what we now know is that babies, when they are born healthy, have a very low rate of micronuclei, okay? No more than 0 to 2 per thousand. However, by the time we're 18 to 25, we've already lost the plot. The DNA damage rate has already increased quite a lot, okay? 2 times to 3 times to 4 times higher, as you can see. And with aging, it keeps on increasing more and more so. Those who live longer seem to be on a lower trajectory. These people here with very high rates of damage probably are dying. And this kind of relationship is shown with other markers of DNA damage. So the question really is there anything we can do, given that we know that increased DNA damage rates increases your risk for developmental and degenerative disease later in life, is there anything we can do to bring the DNA damage rate back down to what we start off in life? right, as a baby, because that seems to be the possible minimum that we could achieve biologically, right? There's always going to be some level of damage. I don't think that's going to be avoided because of stochastic reasons of certain imprecisions in the process of genome maintenance, even under the best conditions. So in a public health sense, I think this way. If increasing DNA damage rate is going to increase the risk of developmental and degenerative diseases, maybe we can think of a different way about public health and start thinking at the fundamental causes of disease, such as genome damage, and ask the question, what is the threshold of DNA damage we should allow? What are the costs of doing that? And can we design exposomes across all aspects of exposures, from psychosocial situation to nutrition to environmental chemicals and so on? Can we design an exposome that enables us to stay below that threshold? Okay. So those are the kinds of questions that is kind of my guiding principle in the kind of work we do. Now, if we take folate deficiency, in fact we can take any nutrient and go into great detail, but why would folate deficiency cause DNA damage? There's a number of reasons. There's a reduction in the SAM-SAR ratio, reduced cytosine methylation. Reduction in cytosine methylation will cause centromere defects. The pericentromeric region has got has got repeated DNA, okay, which is structural, and it has to be well methylated for the pericentromere to function well. Otherwise, it despiralizes. If it despiralizes, the kinetochore cannot assemble, and chromosomes are malsegregated, leading to aneuploidy. That explains why there is an increase in aneuploidy. Of course, we can have abnormal gene expression, and if that affects the DNA repair process, it might result in breaks in DNA from which micronuclei are produced. Elevation of homocysteine can lead to oxidative stress and DNA-based damage and DNA strand breaks. And one of the specific lesions caused by folate deficiency directly is by increasing the uracil to DTTP ratio. Right? Thymidine is actually a methylated form of uracil. You could argue that thymidine is a measure of methylation, okay, just like 5-methyl-cytosine. So if you are deficient in folate, uracil increases. There are glycosylases that try to take uracil out, and in doing so leave breaks. So if you are chronically folate deficient, you will get transient breaks accumulating. And these breaks can lead to chromosomal instability, telomere dysfunction. Later I show some data about mitochondrial DNA deletions that may cause p53 dysfunction and also G4 DNA accumulation. Again, so coming back now to this question, well, if I am slightly folate deficient, does it matter? How big is the effect? So we do a lot of modeling in vitro with human lymphocytes. So we use human lymphocytes, are really good to use because you can study them in vitro as you do measure them in vivo. And as long as you're using, the challenge is to have physiological culture medium that reflects the in vivo situation. Now, what we did here was we cultured the lymphocytes within the physiological range that is considered to be adequate to prevent anemia, between 12 and up to 120 nanomolar, okay? And the question was, well, if 12 is sufficient to prevent anemia, we're assuming it's going to be sufficient to prevent DNA damage as well. Is that right? Is that correct? Is that what happens? That is not the case, because what you can see here, that the DNA damage level at 12 nanomolar folate is about 28. If you take it up to 60 nanomolar, it's about 11. So it's about 3 times lower, let's say. Furthermore, the damage increase that you see at 12 nanomolar folate is equivalent to that of 20 rad of X-rays, which is about 10 times the annual exposure limit allowed for the general population. So if we use the same rationale that we use for radiation, it would be illegal to have people running around with 12 nanomolar folate, right? Using the same. It's DNA damage, it's the same similar type of DNA damage at the chromosomal level, and so on. The problem is this is not occurring just because of one micronutrient. This kind of damage is occurring over a range of micronutrients. We're only looking at one. Okay, so What other micronutrients affect the DNA damage rate for micronuclei bridges, nucleoplasmic bridges? Also, you can use the COMET assay. Here's a study in Brazil looking at iron intake in children and neonates, and you can see here this relationship, that the intake of iron affects the DNA damage rate. Okay? It could be a U-shaped curve for some of the markers, some of them it's linear, some of them a plateau within the range seen. So you can tell by this means, at least through an intervention, whether there is some deficiency that might correct or lower the rate of damage. Another example is zinc. So here's an in vitro study results of zinc, where we used the whole cytome, looking at micronuclei bridges and buds, necrosis, apoptosis, and we also used the COMET assay, plus some gene expression using a proteomic approach. And what you can see is this very sensitive system detecting over a small concentration range, 0 to 16 to 32 micromolar, an effect on lowering the micronucleus index, bridges and buds. And too much starts to increase the damage. And with zinc, there's a very, very tight concentration range. The same with necrosis and apoptosis and with the COMET assay, but you can see here the sensitivity with the micronuclei and bridges is particularly strong. And this is typical of what we see. You can also look at the protein markers, such as gamma H2AX, being very high in deficiency. OGG1 increased because of increased oxoguanine. This is the procaspase that tells you about apoptotic events, and metallothionein, which is a marker of increased zinc. Now, we then went one step further and we did an intervention with zinc carnosine, which is a bioavailable form of zinc. Okay? And what we did is we screened older people for their level of zinc, just their plasma level of zinc, and we selected those that had a lower level of zinc, the bottom half of the number we screened, and we intervened with zinc carnosine. The zinc status increased, metallothionein level increased during the intervention, ZIP1, which is one of the transporter genes for zinc, also increased. We also saw significant reduction in the micronucleus index. In the case of the COMET assay, we did not. What happened was that the COMET assay measure actually increased in the placebo for some reason we don't understand, but it did not increase in the supplemented group. Okay. By and large, what this study is telling us, it is plausible that zinc might be contributing to the DNA damage levels we see in our population, and that to some degree that level of damage could be attenuated or reduced by an appropriate intervention with zinc carnosine. Zinc carnosine was actually originally developed to prevent, to treat barrier function in the gut. So it may have actually applications not only for improving DNA damage rates, but also ameliorating barrier function in the gut. And barrier function in the gut itself may be contributing to the DNA damage if there is leakage of toxins from the gut. Now again, we wrote a review about this. Razina Sharif was the student then, explaining all the possible mechanisms. I won't go through the details here, but essentially the kind of markers you can measure are the transporters, the ZIP and ZNT transporters. You can measure also some nutrient sensing and transcription proteins, such as MTF1 here, which can sense the zinc in the cell and then can induce the transcription of metallothionein. This functions well in normal cells, but not in cancer cells. Now, what about foods and other nutrients? So let's think more broadly now. Okay? Instead of looking at single nutrients, let's go a bit further. So what we did was we did a cross-sectional study in this case where we looked at dietary intake of foods. And this is in healthy people in South Australia. Okay? And what we did was we then analyzed the data to identify which micronutrients from food were associated with reduced damage in the highest and medium tertile of intake relative to the lowest tertile. So what you are seeing, this green line is the lowest tertile reference point, and the gray and the purple lines are the medium and higher tertile. What we observed was a reduction, an association of a reduction with the micronucleus index for vitamin E, calcium, folate, retinol, and nicotinic acid, and for the medium tertile of beta-carotene. However, for the higher tertile of beta-carotene and for the medium and higher tertiles of riboflavin, pantothenate, and biotin, we observe an increase. So this is tending to tell us these markers are probably more associated with flesh food intake, but it also tells us here about beta-carotene maybe not being beneficial at higher doses of intakes. There are also studies with other markers, such as the HPRT mutation assay, which is also measured in lymphocytes. That was done by another group in Northern Europe, also showing this observation that we see here. Now, the reason is not so clear, but there is some evidence that retinol, beta-carotene, when oxidized, can be converted to a genotoxic form that might contribute to this damage, whilst retinol does not oxidize easily, I believe. Now, here's the vegetarian and non-vegetarian study, which actually got us onto studying the B vitamins more closely. So this was one of the very first studies I did at the time. There was a lot of interest on antioxidants. So my boss actually was interested in antioxidants, was one of the reasons also to look at this, because we reasoned that maybe vegetarians have got a higher antioxidant intake relative to non-vegetarians. However, our non-vegetarians are actually healthy, so they took care of themselves. What we observed, however, was this. Essentially, so this is the data in vegetarians and non-vegetarians for micronuclei, yeah, okay, in males and then in females. And you can see the increase with age very easily, from 20 to 40 to 61 to 90. What we observed mainly is that in middle age, the vegetarians are doing better. They have a lower rate of damage. The younger ones, there's not much difference. In fact, the males are doing worse, being vegetarian. And that was because the males were not, in this case, were not taking vitamin B12 supplements, which they are recommended to do if you take up a vegan-vegetarian. They were vegan and they were not taking supplements. Yeah. Okay, so what about lifestyle? You can use this health promotion index. This is work by Morimoto's group in terms of the number of cigarettes you smoke, alcohol drinking, exercise, sleeping hours, and you get a score. A high score of the HPI index means you've got better lifestyle habits. And what they observed here is this negative correlation. So a higher lifestyle score, lower micronuclei, less DNA damage. However, in regression analysis, what appeared was that the number of hours you slept, in other words, whether you slept enough, whether you exercised, whether you worked normal hours rather than too much, right? So as researchers, we break all of these habits, as you know, and whether you have nutritional balance. The point was, if you scored well, you did across these, you did particularly well, and their beta values in the regression were about the same. So nutrition alone is not the answer, right? It is part of the answer. Very important that we recognize this, and that we should think of these other aspects at the same time while we are giving nutritional advice to really optimize the outcome. Now I'm going to switch to telomeres, because telomeres are, of course, the other biomarker which is very well validated, and there's a lot of data in terms of now it's improved actually. It wasn't so good in terms of intervention. There was very little intervention data, but that's improved quite a lot in the past 7 years since I last wrote a review regarding the validation status. But there are clearly many studies associating with disease, with nutrition, now some intervention studies, and also prospectively. So, you know, most of you will certainly know that telomeres occur at the ends of chromosomes. They're a TTA, GGG repeat sequence. And when they become too short, it seems to cause a problem in terms of chromosome dysfunction. The reason you have telomeres is so that the ends of chromosomes are not recognized like DNA strand breaks. Because if they are seen as DNA strand breaks, the homologous recombination pathway will come along and start joining them together. And you'll have absolute chaos because you'll have all your chromosomes stuck together, which you can't, now that's what happens, which you can't segregate, right? So that's the real problem. That's why telomeres are so important and why they cannot be critically short, okay? However, they can be too long and dysfunctional, and you still get the same problem. Okay, so very quickly, the reason why you need telomeres to be adequately long is because if the telomere end needs to form this T-loop structure. And on this T-loop structure, the sheltering proteins are assembled to protect it, to maintain it, and so on. Okay? So you don't necessarily have to lose all your telomere sequence. You just need to lose that it's that short that you cannot make the loop. Okay? Now, there are many, of course, telomere shortening is associated with accelerated aging, liver fibrosis, anemia, immune dysfunction, and other conditions, and as associated with increased risk for cancer. We know it's associated with the dementia as well. These are some of the studies we've done on telomere length and Alzheimer's disease, younger and older relative to controls. It's diminished in lymphocytes, as well as in buccal cells. A lot of this work we also do in buccal cells, but there isn't time to talk about it. And this is a very important development because buccal cells are easy to get, minimally invasively, very easy to store and investigate. But they're not as well validated at this point in time, but they do correlate what we see in lymphocytes. Now, there's a telomere damage cascade. This is in this review we published some years ago, that these effects such as oxidative stress, folate deficiency, niacin deficiency can all contribute through different mechanisms to cause breaks in the telomere, reduced binding of sheltering proteins like TRF1/2, and the loss of telomere length control, which is due to inappropriate or inadequate tankerase activity. And this leads to dysfunctional and or short, long or short telomeres and telomere end fusions. Now, the interesting thing about telomeres is that the damage in telomeres is not repaired. And that is another reason why having too long a telomere may not be such a good thing, because it becomes a bigger target for damage. The problem here is this persistent, when there is this persistent damage, it creates what's called the persistent DNA damage response, which stimulates cellular senescence and aging. Now, if everything worked well, cellular senescence is a good thing. As Judith Campisi indicates in her review in Annual Reviews of Physiology. It's a very nice paper to read. So on the bright side, this persistent DNA damage response is a signal. There are chemokines that come out of the cell, encouraging the immune system to take the cell out, to remove it. But as you get older, the immune function doesn't work well enough or efficiently enough to remove this. So then you remain with this pro-inflammatory condition staying and increasing with age, increasing inflammation, excessive cell proliferation, DNA damage, stimulating carcinogenesis, and so on. So this is thought to be part of the inflammation scenario. So others are trying to consider what can you do about this. Maybe we should try and help the senescence system to do its job. Okay, so this is a lot of research going on in this particular space. We do quite a lot of telomere work now. This is a study we did in children, which I think I showed this maybe yesterday or the day before, showing that there's a negative correlation with zinc. So too much zinc may not be a good thing for telomeres. And also there's an association with the reduced folate carrier AATA. So homozygotes have got shorter telomeres. And we know that this is also associated with more micronuclei, as I show a bit later. And from this study in Spain, a higher intake of fruits and vegetables and less white bread is associated with longer telomeres. So we know that nutrition is affecting telomeres in children. And in adults, there is actually a host, and there's quite a lot of information. A lot of these, though, are single studies. except for the Mediterranean diet. The Mediterranean diet has been studied about 5 times, and consistently telomeres are longer in those with a higher Mediterranean diet score. So we can be a bit more confident about that. But overall, we know that telomeres tend to be longer consuming the Mediterranean diet, the prudent Korean diet, higher dietary antioxidants, vitamin C, E fiber, higher serum vitamin C, D, carotenoids, calcium magnesium ratio, higher intracellular zinc, gamma omega-3 fatty acid supplementation intake, caloric restriction, and multivitamin use. There's, on the other side, shorter. There's also a study showing multivitamin use associated with shorter telomeres. So it depends in which context and what kind of multivitamin it was. Higher dietary intake of red and processed meat associated with shorter, white bread, sweetened soda drinks, short, medium chain saturated fatty acids, higher plasma calcium, magnesium, zinc, higher homocysteine, that is quite consistent as well, and the lowest and highest quintiles of serum folate. There is apparently, again, a U-shaped relationship here. And obesity. There are some intervention studies. This is one we did ourselves in overweight, obese men who were either on a high protein or a high carbohydrate weight loss diet. These men were absolute heroes because our physician researcher managed to convince them to donate rectal biopsies 3 times, mid-rectal biopsies. And they said, what can you do with them? We're scratching our heads. We said, well, let's try telomere length, because there wasn't much to work with anyway, which we did. And what you can see here is an apparent restitution of telomere length in the rectal biopsies, occurring both with the high protein or the higher carbohydrate weight loss diets. This study here is an intervention in prostate cancer patients who are in a watchful waiting situation, where they were opting they would rather not be treated at this point in time. And this is work done by Dean Ornish together with Elizabeth Blackburn. And in their first paper, they observed that adopting this lifestyle that Dean Ornish promotes, with a low-fat, whole food, plant-based diet, with moderate aerobic exercise, some stress management, some meditation, about 60 minutes a day, supplementation with soy, servings of tofu, vitamin E, selenium, and so on. And in the first paper, they observed an increase in telomerase, which they reported in the Lancet. And again, in the Lancet, some years later, they reported the effect on telomere length, which was apparently longer, but not by a great margin. It certainly was longer, but it wasn't that much longer. Mitochondria. Why are mitochondria important? Well, mitochondria generate the ATP we need to keep all of these together, all of these systems together. Nutrient deficiency can affect mitochondrial DNA damage and the mitochondrial copy number. If there's increased damage, there's more reactive oxygen species, which can accelerate aging. These conditions, Huntington, mutant Parkinson's disease, ALS, and Alzheimer's disease, all contribute to mitochondrial dysfunction. In the case of Alzheimer's disease, A-beta-42 can block TOM. TOM is a structure on the membrane of the mitochondrion, which allows the import of nuclear encoded proteins that the mitochondrion needs for its electron transport chain. And the same problem can happen with ALS. So the mutant SOD in ALS can block TOM as well. So these are some of the important issues and mechanisms affecting these conditions. Now, it's known that mitochondrial DNA deletions, this was the first paper reporting this, increases with aging in non-dividing tissues, such as here in this case the brain, but also in the muscle. This was shown, the muscle was shown, studies were shown by Tony Linnane in Australia. This study here is interesting. This is a study done in Taiwan by Wei-Fen Huang, which has actually attracted my attention a lot. She had been studying the impact of folate deficiency on mitochondrial DNA deletions. So you've got these major deletions that can be induced, that appear to occur spontaneously. But what she observed was that these deletions in the mouse model were actually induced by folate deficiency, and to a great extent across all tissues, as shown here. She plotted the data like this as a linear relationship, but you could also plot it as a hockey stick. And you can see here a steep decline in mitochondrial DNA deletions and then a plateau if you want to observe or interpret the data in that manner. The very interesting point was that the lymphocyte, the change in the mitochondrial DNA in the lymphocytes, the increase in mitochondrial DNA deletions correlated very well with that in the brain, correlation factor of 0.9. And these and also with folate status, correlation factor of minus 0.7. So this provides us with some hope. And this is not a very hardly researched area. We're working in this. We're starting to get some resources to be able to do it. But I think it's a very important area to look at. Not just the nuclear genome, but also the mitochondrial genome. Okay, so quickly now. So what are we doing about translating into practice? So these ideas and concepts, given that we now have very large databases for micronuclei and telomere length, we can diagnose the micronucleus frequency and telomere status of individuals and classify them as to whether they are, in the case of micronuclei, low, medium, or high, and the same with telomeres. But of course, with telomeres, you want them to be longer, and micronuclei, you want them to be lower. And in this way, you can do this diagnostics. It's available now to the general public. We launched the first Genome Health Clinic about 8 years ago. It's called Reach 100. There are other telomere-related kind of clinics like this that can provide a service. The concept that I wrote about some years ago is described here, using automated high-content diagnostics to measure genome health, nutrient status, and genotype, lifestyle genomics, information as an expert advice system to provide appropriate validated advice based on the results that are produced, to provide individualized diet and lifestyle, and to optimize genome health and reduce the risk of developmental degenerative diseases. So that's the concept in a nutshell. Part of this approach is also to look at the genetic side. Now, I won't spend a lot of time. I'll just give some examples, because the time right now is zero, of the knowledge about genotype affecting these DNA damage markers is increasing. I'll show quickly some examples. In ataxia telangiectasia, there is a study here done in the mouse model showing that if the mother is treated with N-acetylcysteine, which is an antioxidant, the fetus, the level of oxidized guanine in the fetus is normalized compared to the non-mutant via the mother, right? So this is very interesting. And in the field of ataxia telangiectasia, they are trying to do these kinds of interventions. Actually, I just came across a paper for families in Australia where they show the families and the kids who have this condition are actually not, their diets are really bad. So they're probably aggravating what is already a bad situation. So we can personalize nutrition in these cases. Another bigger case would be Down syndrome, where there's an accelerated aging syndrome with with the biochemistry is well known, and it's possible to intervene. This, I think, is tractable. It's possible to do. This is our own study here looking at folate polymorphisms. We did not see an effect of MTHFR, but we saw an association with the A276G MTR, methionine synthase, and the reduced folate carrier AATG polymorphism. Here's a study in China showing, this is though an in vitro model, showing that micronuclei are increased in those who have breast cancer status, but can be reduced by vitamin B6 intervention. And interestingly, in the model, also showed that if you look at the polymorphism of serine hydroxymethyl-transferase, which is the enzyme you need to convert 5,10-methylene tetrahydrofolate to 5,10-methylene tetrahydrofolate, the form needed for thymidine synthesis, then the damage is modulated by polymorphism in that SNP, in a SNP of SHMT, which requires vitamin B6 as the cofactor. Okay? So you can, by improving vitamin B6 status, correct or normalize the damage in these cases. This is a study in relation to vitamin B6 and the polymorphism in the MAT1A. done in the Puerto Rican study, showing that a particular genotype benefits whilst another one doesn't in the MAT1A. This is a study on PPAR-GC1A, again showing an association here in diabetes and cardiovascular disease of this polymorphism with DNA damage, in the case oxoguanine. Here's another study published this year on telomere length and IL-6 levels being associated with each other with this polymorphism in an obesity treatment intervention in young adolescents who are obese. And here is in fact a study in the famous Predimed Navarra intervention showing that this polymorphism, Pro1,2 ALA polymorphism of the PPAR gamma 2 gene predicts who benefits from this intervention. So, is this the restaurant of the future? Considering your genetic profile, I recommend the fish of the day with soybeans for main course and only 1 glass of wine, berries for dessert, and a strong coffee to finish. Well, maybe, maybe not. But we do know that restaurants are starting to ask that question and inquiring to us about providing personalized diets and approaches to their customers. Okay, that's it. Sorry, I'm a bit late.